Thank you to the organizers. This is a really wonderful meeting for me. I do methods development for computational biology and computational genomics, and this is what this meeting's about. The organizers instructed us that we can take up to 20 minutes to introduce ourselves, so I thought take full advantage of this. Tell you that I'm recently moved to Caltech, so I'm kind of local, and plan to be here most of the days this week and get to know a lot of you. So I thought I would talk today about pseudo alignment, and this is a research topic that I'm involved and engaged in. And I chose it partly because I think there are some lessons about the field that should be useful to those of you who are just starting out, and to those of you who've also been doing this kind of thing for a while. And what I start with is, first of all, tell you the papers that I'll be mentioning or talking about. I won't go through all of these papers, but I'll mention some of them. And if you want to follow up after the talk, then these references will be useful for you. The main idea of pseudo-alignment that I'll discuss is from a paper published last year. The idea is primarily that of my former student, Nicholas Bray, so I've bold-faced him. And it was a project that was done together with Harold Pimentel, who's here at CGSI, so you should also get to know him. As well as a collaborator, Paul Melstead, who's at the University of Iceland. I've also been collaborating with David Che, who's another speaker and participant at the CGSI this week, so please also talk to him. We've been applying the idea of pseudo-alignment to single-cell RNA-seq, and that's another theme that we'll run this week, so I'm going to try to touch on that briefly. The third reference is an application of this recently published to very large-scale analysis. And one of the themes that's emerged in computational biology over the last few years is that of reproducibility and computations at scale. And I think that's a very nice paper. It's not ours, but I think you'd benefit from knowing about it and reading it. And finally, I'm going to mention a very classic paper that most people have never read, but it's sort of the origins of our whole field. So I thought I would, and it's related to what I'll talk about, so I'm going to mention it. And that's the paper by Peter Sellers. From 1972, it's actually interesting for me. You know, a lot of people here, I imagine, are from different academic backgrounds. And my academic background is from combinatorics and mathematics. And this particular paper was published in the Journal of Combinatorics, which is the premier, really one of the premier journals in combinatorics. And it discusses basically the sequence alignment problem, which you all probably also have already heard about or seen if you've taken any bioinformatics course these days. At the undergrad level, you learn how to align two sequences. And one of the things you learn is how to do it in running time n times m, where the sequences have length n and m. And first of all, they switched m and n here, which is funny. But anyway, the running time of this algorithm is m squared n, which is actually really suboptimal. So you can get published in math journals with suboptimal algorithms. That's one lesson. So it's not a bad idea. But anyway, I mentioned this work because it's about sequence alignment, and I'm going to talk a bit about read alignment. And the two sound very much alike, but not just to students, but also to the experts, and they confuse them. And actually, one of the insights I want to discuss in my little talk today is why it's kind of useful to take the time to think about things and understand sometimes how they're different. This paper by Peter Sellers, it was really about what's called the Needleman-Wunsch algorithm, which you've heard about, which was published 2 years earlier, but in a different journal, in biology, in a biology journal. And the mathematicians didn't read biology journals. Biologists weren't reading math journals at the time, so they didn't know about each other. But in the Peter Sellers paper, which had a huge influence on the actual paper that ended up being really important, the Smith-Waterman paper, I'll get to that in a second, they realized that this was going to be useful for looking at evolutionary distances and applying this to alignment of biological sequences. And in fact, that's what they did. this was actually shortly after the time when people for the very first time actually sequenced anything and did the very first alignments. And you may or may not know that this was from a paper of Linus Pauling and Emil Zuckerkandl from 1963, and I like to show it off now because it's from Caltech, which is where I'm now at. They sequenced amino acid sequences, and in fact, Peter Sellers in his paper applied their algorithms to aligning amino acids. But what I wanted to say in one slide is there is a really important underlying principle to doing these kind of alignments. These are just 4 amino acids, and that is that the alignment is somehow a biological problem. And what I mean by that is that while the mathematicians and computer scientists like to talk about getting lots of matches, like M and M here, and not so many mismatches, and minimizing these sort of insertions or deletions, underlying this alignment are little phylogenetic trees, because there's a presumption that these sequences had a shared ancestor and evolved by some evolutionary process that included mutation or deletion or insertion. And ultimately, this representation and the algorithm to produce this is reflecting modeling and knowledge of this evolutionary process. There's a second issue to doing sequence alignments, which is very nicely discussed in a paper by Gert and Lunter, which I urge you to look up, from 2008. In this paper, he discusses how sequence alignment is also fundamentally a statistical problem, and he gives examples of things that may or may go wrong, where maybe this is the correct alignment, but a program might produce this alignment because of the model that's being used to do the alignment. And so it doesn't say it on the slide, but really what this is about is model selection in statistics, which goes hand in hand with the evolutionary process in biology. And actually, one of the great things about doing and solving problems like sequence alignment is the models sharpen the biological questions. So I would argue that sequence alignment was inherently a biological and a statistical problem. But I want to say 3 final things about it. And then I'll get to what I am actually going to talk about. The first is that problem formulation was key. And I don't have time today to go through the whole history, but what really ended up mattering for sequence alignment was the Smith-Waterman algorithm that was actually published in the 80s. And that solves the problem of local alignment. That's a seemingly different and much harder problem. And the key insight of Smith and Waterman was to realize that you could use the algorithm of Needleman, Wunsch, or Peter Sellers, or David Sankoff. A lot of these people came up eventually with the same algorithm by simply adding a 0 in some key spot in the algorithm. It's a really beautiful thing. And I'm not going to discuss it, but the problem was more complicated. Everything was more complicated, but understanding, A, what problem you ought to be working on was the key to then realizing that actually solving it is really straightforward. The other thing I wanted to say is that sequence alignment was figured out a long time ago. And basically, already back then, other than the mathematicians, everybody got the fact that it was quadratic in the length of the sequences. And it turns out, recently proved just 2 years ago, that that's basically the best you can do. So the optimal algorithm was actually figured out early on. And in some sense, the computer science has not been the challenge, even though many people have worked on this since. Challenge has been getting the biology and the statistics that complement it correctly. So I want to talk about a different problem today, which is the read alignment problem. But I want to give it a different name because I'm going to try to emphasize that it's fundamentally a different question, even though its origins are with sequence alignment, and therefore people evolved the same methods, even though they may not have been the most suitable. Actually, very, very first read alignments were done in the late 90s. This is a screenshot from a program that was part of a suite of tools developed by another mathematician, at the University of Washington. And these tools were used to correct errors in assemblies. I think this is from the suite of what's called FRAP. And those of you who are young here probably have never heard of that. So I'm going to tell you about what it did. So this very first program was being used to take fragments of DNA coming out of sequencers at the time, Sanger sequencing reads. They were about 700 base pairs long, and aligning them actually to assembled contigs. These were sequences that had already been assembled partially from these very same reads, but for the purpose of figuring out where in these sequences there may be errors in the assembly. Because if you could pile on all these reads onto one of these sequences, you could go and see, oh, maybe over here, a lot of the reads are saying C, but I, for whatever reason, assembled an A. It was a kind of a form of error correction. In fact, this was called consensus mapping. And so there's sort of two components to this, and I'm just going to give them symbols. So the very first part is sort of constructing this map, which says, for each of these fragments, which of these targets does it belong to? And I've added in the empty set because some of these fragments were not assemblable, and they actually didn't originate from any of the contigs. And so the second part of the map involved actually figuring out where on an actual sequence this fragment literally actually originated from. And I'm going to give that another word just to conceptually in your mind distinguish these two concepts. So the output of what was called a read alignment was literally what looked like a sequence alignment, the kind that people have been doing for evolutionarily evolved sequences, where you have the same kind of matching and mismatching. But unlike that setting, where you were looking at maybe a human and mouse sequence or different globin sequences in the case of polling from the same organism, but still related to each other evolutionarily, here, these two sequences are literally the same. It's just that this one was produced from an assembly, whereas this one just came out of the sequencer. So this is sort of actually a very different thing. It's an alignment of technology, not biology, right? And so it actually is a different thing. But in the original FRAP algorithm, the way this was done was by adapting the sequence alignment algorithm of Smith and Waterman, which again I didn't talk about, but it's the algorithm that just was really developed for aligning sequences. So not to belabor the point, but that's how it was done. And I just wanted to show you that back in those days, there was also a large gap between statistics and computer science. And actually, that's one reason I really love this meeting, and I thank the organizers for putting it together, because this audience is very diverse, and its goal is to bring different ideas from one area to another, and hopefully for you who are coming from different backgrounds and different interests, to talk to each other. So the metagenomics people should talk to the statistical geneticists. Well, this is a great example. FRAP initially had a score of 11 for a match, -2 for a mismatch. And this should strike you as strange. For one thing, these numbers are integers, and there's no particular reason they ought to be other than math people like integers, right? So it took a long time to actually make these things more statistical. So what happened is that algorithm FRAP, it was like 1998, I think. But then in 2003, when some of you, I guess, were born, something like that, we started getting like really a lot more sequencing happening. And this is a slide from a former student of mine, Atif Rahman, who just got his PhD 2 years ago. And you've seen many slides like this. I really like this one. You can't read very well here, but there's all the different technologies are represented by circles here. This is time. On the y-axis, you have cost, as in the number of mega base pairs of sequence that you can sequence for $1,000. So as you go up on that axis, sequencing is getting cheaper. And the diameter of these circles is proportional to the read length. you can see that back here, Sanger sequencing had pretty long read lengths. Now we're going back there. And the actual boundary here is somehow proportional to the error rate. So you can see that with time, and we all know this, sequencing has gotten a lot cheaper, and the reads have gotten better, and so forth and so on. But all of this technology has driven the need for read alignment, okay, as opposed to sequence alignment. In the applications that come out of these technologies, there's sort of a different question than what Frapp was addressing, but similar in its nature. Goal is now to take a read, again, identify where it comes from different targets. These targets might be DNA sequences, they might be RNA transcripts, but the goal is to count, and that's why these are called sequence census methods, the number of reads originating from these different targets. So this has become known as the read alignment problem, and it's really, in some unfortunate way, actually, somehow made the reads, the sequence alignment problem kind of uncool and unpopular, and everybody stopped working on that problem. And everybody now works on this problem, okay? But they're both actually really important and different. So Nick Bray, who I mentioned previously, had an observation a couple of years ago during his PhD work, which is that he was just thinking about this sort of from scratch and thinking about the problem formulation, and realized that people were adjusting and adapting sequence alignment methods for a problem that was fundamentally different and therefore maybe required a completely different problem formulation. In particular, what he realized is that for sequence census, when you're counting and doing an assay such as RNA-seq, you only care about what we then called pseudo-alignment, which is really which target your read comes from and not where it aligns. Because with read alignment, these days, we're not correcting so much errors in assembly anymore, we're just counting those different targets. And I wanted to just give just tell a little anecdote of why I think he had this insight, was that Nick actually worked with me as an undergraduate, and around 2001 to 2002, he worked on sequence alignment and genome alignment. So he had that area really figured out in his head. And you know, that actually really, I think, helped him when he was looking at this new problem to see the distinction. So it's sort of really useful to, you know, when you're switching areas, to not, you know, forget everything you learned before because it's no longer useful. It's actually very much the opposite. And so that's sort of what led him to the idea. And from the sort of formal point of view, this idea is trivial and is a bit embarrassing for me to present as a research talk at the CGSI, because really what he was saying was, you know what, let's only worry about this part. Let's just forget the alignment part. Okay, well, that's kind of trivial. So what? Okay. So I'll explain what the big deal is, because it is conceptually very simple. It's algorithmically very simple. But I think the problem formulation was not simple. And that's, I think, what the contribution here was. But in the spirit of the CGSI and being able to talk to people across fields, I want to go through this carefully and slowly. So let me repeat. So in alignment-based analysis, you have, in this toy example, 5 reads that have an actual sequence. Here I've put 3 different targets, and you are trying to find exactly where this goes, the sequence GGG, TTT, exactly which position in which target. Really straightforward stuff. Okay? So in a typical alignment, sometimes you can identify that uniquely, but sometimes you can only identify it ambiguously, and that happens for a number of reasons. In higher eukaryotes, at least if you're doing an assay such as RNA-seq, there's alternative splicing, and so you have multiple isoforms of the same gene, which are very repetitive. You also have, in general, large gene duplications often that leave you with very repetitive sequence. Or if you're doing an application such as metagenomics, where this all applies as well, these targets are different genomes of bacteria, which might be of the same species. Maybe they're different strains, so they may be very similar as well. So sometimes you get a unique hit, sometimes you get ambiguous hits, and a lot of tools have been developed to solve this problem of finding these hits. But even when they call those tools ultra fast, they're very slow. And I'll show you that that sort of has a number of implications about accuracy as well. A lot of the times, we don't really care about this information. If you're doing metagenomics, we just want to know the content of the sample, maybe. We don't really care about this read at all. Who cares about the read? In fact, their labels are some numbers in hex or whatever. They're not actually interesting in of themselves. What's interesting is ultimately quantifying the samples. So what I will not do in my talk today, but I wish I had time if I was giving a very long tutorial about this, is tell you first how people actually do or did read alignment for the last decade or so, because that did not turn out to be a lot about biology or statistics, but a lot about computer science. And there's a lot of interesting tricks. The first and foremost one was the Burroughs-Wheeler transform, which I've shown here, which involves cyclically permuting the sequences and then recording the final column. But I won't do that today. I'm going to go back to the pseudo-alignment and explain that what you want to actually achieve at the end of the day is simply record this data instead of all this detailed sequence by position. Now, what is this data? Well, S here stands for sets now. And if you look here, there are actually a number of different sets. There's the set T1, T2, T3, which appears twice here. There's the set T1 by itself, T3 by itself, and T1, T2 by itself. And those are the 5 sets here. So this is the one that appears twice. And then you have these 3 sets. And some sets appear 0 times. And there's going to be others of these. Okay? What you want to do is simply quickly arrive at how many times did we see a read that was ambiguous to all 3, a read that was unique to this one, unique to that one, or maybe unique to the second one. Okay? And so the idea is to compute that directly. And this was our little trick to doing it. And we implemented it in a program called Callisto, which I've labeled for this slide because there are other ways to do this. So now I'm going to show you an algorithm to do what I'm calling pseudo-alignment, which is to produce just this data, but it's a very specific algorithm, so I've given it a name. And there are other ways to do this as well. And maybe some of you will be very clever and find new ways to do it. It's a pretty new problem, so not that many people have thought about it. So what we did is we built the Bruijn graph. This is a graph that's usually made for building assembly reads. And you'll hear more about that this week in some of the talks about sequence assembly. We sort of co-opted this idea for another purpose. Instead of assembling the reads, what we're doing here is assembling the actual targets that we know ahead of time. So it's sort of reversing the role of the Bruijn graph in the first place. So in this little toy example here, there are 3 targets. They're labeled pink, blue, and green. And we've simply taken their entire sequences. We've chopped them into little bits of length k. And k in our actual program is 31. When we do the human genome, it can be shorter if you do a smaller genome. But again, we're not chopping up the reads. We're chopping up these sequences. And each little circle here is a k-mer. And then we're decorating this graph with the information on which of these sequences it actually came from. So for example, this k-mer right here was present in all 3 of these sequences. So we give it the label green, pink, and blue. We can actually be a little fancier and record also where in these sequences it was located. But we're going to use that decorative information to rapidly compute the pseudo-alignment. We're never actually going to use it to do a complete alignment because we just don't care about that. So building these Bruijn graphs is actually a very tractable problem and very quick to do. In fact, for the human transcriptome, this takes about 10 minutes. Thanks to all the technology developed for sequence assembly. So actually, that's another meta point of my little research talk is sometimes people nowadays say, sequence assembly, that's kind of a solved problem. I mean, who cares? Well, actually, there's many reasons that's not true to begin with. But another point is that that technology for sequence assembly is actually going to be useful for other things. And this was one example. People hadn't figured out how to do all this Bruijn graph stuff very quickly. We wouldn't have been able to do this project because we really leveraged a lot of tricks of that trade. but now co-opting it for a different application. So, well, how do you use this to do pseudo-alignment? Well, this is the sort of embarrassingly simplest part of the talk. Well, you go now and you look up your k-mers, and you look up their colors. Now, one of the things is people had actually done this before, taking k-mers from the reads and noting which targets they were compatible with. And then actually metagenomics, that's a common method called alignment-free analysis. But actually, we're not going to do that. We're not going to do alignment-free analysis. We're going to then couple all that information together, and that's what makes this require a new word rather than just calling this alignment-free analysis. Specifically, what we do is we intersect all those classes. So this k-mer is compatible with these 3 targets and so on. But then this k-mer over here is compatible only with these 2. And so we intersect all these as sets because we're interested in the pseudo-alignment of the entire read. We don't care about one of the k-mers, really. They're just an intermediate object for us to utilize. We care about the whole read. And if these k-mers over here are compatible only with these 2, then even though these k-mers were compatible with all 3, we know that as a whole read, it couldn't have been from green. It's really, that's the whole idea. I mean, we got a paper out of this, actually. It's great. So we do these intersections, and that's the output of the pseudo-alignment. But we've lost something here. We've lost, a detailed alignment. For example, if there was an insertion, or let's say, there was a mutation due to the sequencing error, so I shouldn't call it a mutation, let's just call it a mismatch, we won't find this k-mer at all. It'll not even map to this graph, so we just ignore that. We actually might not find a whole set of k-mers, and that's fine. So we're not going to get out a detailed alignment, but we get out this set. And if we want to spend a little bit more energy, we can maybe not go the whole way towards an alignment, but we could find out where this was located so that we at least know where the read alignment began. Okay? So that's not a complete alignment. We still call it a pseudo-alignment, but it's a little bit more information. So, yes? If you get an empty set. You can. That's a very good question. Sorry, let me go back there real quick. So the question was, sometimes you might get an empty set. For example, if this k-mer over here, if there was an error in the sequence, maybe it maps to a whole new target that it's not really from. Then you'll get an empty seed, and you fail to pseudo-align your read. But that turns out that happens very, very rarely. And so you lose very few k-mers for that reason. What's very nice about this method, on the other hand, is that usual mismatches simply delete k-mers. And therefore, you're very robust to error, regardless of where it is located in the read. Unlike standard alignment methods, and I won't get into comparisons, which require a seed to align exactly to start things off, here your errors can occur anywhere. But yes, you might sometimes get an empty set. And again, I won't have time to talk about that, but it's another example of problem formulation. Initially, when that happened, we were relieved that it happened rarely. And we thought, OK, it's a kind of a bug of this method. It's not too bad. Then we turned it around and said, you know what, this is a feature. Okay? Why is it a feature? Well, we realized that in cancer data sets where you do cancer RNA-seq, you might have 2 different targets that have fused in the genome. And then we get an empty intersection. So we have a new method now which collects all the reads that have empty intersection that didn't align, and then we go and figure out which parts their separate pieces pseudo-align to. And we have a very rapid method now for identifying cancer fusions. But yeah, so good question. So we call this method Callisto, and I think I have 10 more minutes or 5 more minutes, so yeah, something like that. So I'll wrap up soon. But anyway, we call this method Callisto because k-mers alone lose lots of information. They're strung together only. Okay. There we go. It's also true that Callisto is a bear, and this was developed at UC Berkeley. Now I'm at Caltech, so my next program is going to be named the Beaver. Okay, busy beaver. All right. After you come up with an idea like this, there's a lot of kind of clever things that you can do downstream. Our actual ultimate program leverages a number of downstream tricks, which, for example, if you've already built this structure, let's say you get your read and you identify this k-mer in it, you can sort of, knowing the structure, figure out ahead of time that looking up the next k-mer in your read assuming that this is where the read is coming from, is going to not reveal anything because these intersections are the same. So why bother checking it? So what we actually do is we have a skipping rule where when you look at this k-mer, we've recorded here that there's no point to look over here, so you're going to look 4 k-mers ahead. And so we actually skip frequently across the entire read. We often can get away with pseudo-aligning a read by examining only 2 to 3 k-mers, which is one reason this method is so super fast, because you can rapidly identify the part of the read that's actually informative. You know, most of the read is completely uninteresting and informative. It's just revealing the same information to you. So that's just a really simple trick that sped up our programs by like literally a factor of 8. So and there's probably more to drive than even that trick, and we have a couple others which I won't talk about today, but it's fun to think how to squeeze every last ounce of speed out of it. So what comes out of this method are these counts, and we gave them a name that I'm trying to popularize because it's not common in the literature on read alignment or genomics or single cell this or single cell that, but we feel that it's actually the relevant object to be talking about, and we call these transcript compatibility counts, or TCCs. So what are TCCs? So the TCCs are given all these reads, and given these sets, the multiplicities of the sets. Because that's really the data of the reads. So it's not how many reads aligned to this target anymore. That's not really what the method is telling you. It's telling you, this set occurs twice, 5 times, 1 time, and so on. And we call that a TCC, or transcript compatibility count. We have another word for these sets that came out of literature prior to our work on pseudo-alignment, where these were called equivalence classes. Because there is a notion of equivalence class between reads. Reads that pseudo-align to the same sets, you can think of them as equivalent. They form an equivalence class. So these are called equivalence classes because they're equivalence classes for the reads, and these are called transcript compatibility counts. And again, I don't have time today to talk about it, but we have done theoretical work, in particular by Vasilis Dranos, who's a joint postdoc with myself and David Che, where there's reasons to believe that actually doing a lot of downstream analysis at the level of these TCCs is more accurate than first trying to decompose them into individual counts of the individual targets that comprise them. That can be done. So you can quantify the individual targets so that you can know how much of T1, how much of T2, how much of T3 you had. But when you take the TCCs and try to derive these quantifications, you're inherently introducing some noise because you're assigning these ambiguously pseudo-aligning reads in some statistical way. So this is useful. And I find it kind of interesting sometimes. The field is so ingrained in this mode of thought that there are recently 2 different papers published describing simulation frameworks for single-cell RNA-seq. And what actually is simulated is a table of gene counts by cells, where it's basically, so there's no read simulation, it's just counts, because people are just assuming, this is the data, but it's really not, okay? I would like a single-cell simulator to simulate these counts, okay? Because really that's what's accessible to us. This data has had noise added to it just by virtue of requiring inference. Okay, so one of the things that I like to emphasize when I talk about pseudo-alignment is that it's very fast, but that speed is interesting not just because it's fun to now pseudo-align on your laptop, but because it allows you to do things that wouldn't be possible otherwise. And one of them is to actually quantify the uncertainty that's introduced in the step of taking the TCCs and actually quantifying the targets. So Callisto actually does that, and it does it by using the bootstrap. And I wasn't here last year, but I heard there was a great tutorial about the bootstrap. And this is a good application of it. So what you can do with Callisto, because it's so fast, and because it's so simple, is you can bootstrap the TCC counts, then quantify each time in order to assess the extent to which those estimates of the individual target abundances are uncertain. And one way to think about doing this bootstrap is, for biologists who are here, is it's sort of like you got to go out and do a technical replicate of your experiment for free. Okay, so that's how I like to think about it. So you had your actual data, but now you're going to pretend that you did 3 technical replicates, and in fact, you're going to get 3 different quantifications. And we actually did an analysis in our paper where we took a very large data set and we split it physically into different pieces. And then we compared how the abundance estimates for those different pieces actually compared to what comes out of the bootstrap. And there's a very, very strong correlation. It's not perfect, but it's very strong. The colors here actually have to do with the abundances of the targets. And I won't go through it now, but there's kind of obvious correlation. Things that are more abundant you can be better at estimating your uncertainty in quantifying them, but that relationship is not that simple, actually. So we've also done separate experiments where we've literally taken technical replicates out of an experiment and showed that the bootstrap recapitulates the variance quite well. So it is a kind of good, decent computational proxy for doing a technical replicate. OK, so what I wanted to do next is, and I'll just go a little quickly because I want to end on time, is tell you very briefly that this whole framework works for single-cell analysis, for example, for single-cell RNA-seq. You can repeat the pseudo-alignment, obviously, for different cells. And we've done that. We have a version of the program that's sort of optimized for single-cell RNA-seq, so that instead of having to yourself manually run it on each cell, you can put in all your different cells and do this all at the same time. It's actually kind of interesting where a lot of the literature, for example, in single-cell RNA-seq, but in general, for the kind of sequence census methods, has to do with issues such as how you normalize your data. How do you take your gene counts and normalize them? Well, we want to do the same thing, but we don't have gene counts now. We have TCCs, and we want to stick to these objects because they're more accurate, because we don't want to introduce noise by inference. So if you want research problems to work on, there's a lot of them here, because basically every kind of problem people have worked on for tables or matrices that come from gene counts, you really want to redo with tables where the rows are now sets instead of individual genes. So I won't go through this again, but I wanted to tell you that one of the very nice things about this is that using Callisto to do pseudo-alignment works out to be about 30 seconds. You could also say cents, but that's also true, but about 30 seconds per cell. This is really great, actually, because you can actually analyze data on your laptop instead of requiring a big server. And again, I don't have time today to talk about it, but it allows work to be much more reproducible, and it allows you to ask what-if questions. What if I add this particular gene into my annotation? Well, how does it change my results? And there's a really nice paper recently from Evan Eichler's group. He studies segmental duplications in the human, and they were interested in a specific gene that they identified as a duplicate, which is not in the standard annotation that you get from UC Santa Cruz Ensemble. They were interested in how the whole GTEx is a large consortium that's done RNA-seq, and soon it will be close to 20,000 samples from different tissues in human. They were interested where this particular gene is expressed, so they wanted to reanalyze all of GTEx. And they really couldn't have done that without a tool like this, because GTEx itself took months to just do one single quantification and alignment of the data. So they used Callisto to just build a new index that has this extra gene, quantify all the data, and get the result. And you can also do this with toil that I mentioned initially, but that I haven't discussed today. So I think I'll stop. If you're interested to look at the code, it's up there on the GitHub repo. One of the things I'd encourage you to look at, because it's a principle that was taught to me by my students, specifically my former student Harold Pimentel introduced this into our group, is this idea of doing all our work completely reproducibly. And that's turned out to have great dividends for our research, because we can now experiment with new ideas very quickly. So this paper, this is the code for the program that's associated with the paper I showed you, but there's a separate GitHub repo you'll find from going to that link that reproduces all the results in the paper. And if you're savvy, what you'll do is, and this is totally legal, is you'll take that code and you'll try some tweak and you'll get some new results, and then all our code's free and licensed freely for doing all this reproducibility. So you can just write another paper, and you can have all the figures we had. And it's totally legal, okay? So, okay, think about that. One thing that we're quite proud of is that pseudo-alignment right now is only about 4 or 5 times slower than counting the number of characters in your reads, okay? So we're pretty close to basically being optimal, because you basically are gonna have to read your data, right? I mean, if you don't want to read your data, that's a whole nother, that's meta pseudo-alignment, okay? But let's use the actual data. So we called this method sort of near optimal just because it's seemingly about as good as you can do. And it's literally just about running time of looking at your data. So I'd like to thank Nicholas Bray, Harold Pimentel, and Paul Melstead. Paul is a computer scientist who actually came up with a lot of the ideas of how to use the de Bruijn graph to do this, and did a lot of the implementation to make this very fast. There's some interesting theory to work on, which we haven't really had a chance to do, but I'll throw it out there. There's a question of, given some kind of model for the sequences of the targets. So this is not evolutionary models now. These are models for what the target genomic sequences or transcriptomic sequences look like. What are the guarantees for the alignment? For example, Eran noted that if some k-mers don't align, you might get empty intersections. Can you give guarantees on how frequently that'll happen? We don't have such real theoretical guarantees right now. Our work is empirical, is very good, but I think it'd be interesting to do that. I think I'll stop there and take any questions. Thank you very much.